Welcome to this webcast, in which I will explain the fundamentals of our myeloid cell therapy concept, which represents a platform for personalized medicine in a variety of inflammatory disease settings. In the second picture, the clinical treatment challenge, you can see a cartoon in which the blue area represents a tissue mass, be it the nerves in the brain, the cartilage in the joint, or beta cells in the pancreas. At the start of life, represented on the far left of the diagram, the tissue integrity is 100%. In many disease settings, tissue becomes lost as a result of inflammatory immune processes, which is represented by the diminishing blue area. In many diseases, there is excessive loss of tissue until a stage is reached when the individual exhibits clinical symptoms and receives a clinical diagnosis, herein represented by the arrow and the red part of the figure. So at this stage, a majority of the beta cells in the pancreas, for example, have already been destroyed when a diagnosis of type 1 diabetes is given. Similar situations are evident during the secondary progressive phase of multiple sclerosis or in Alzheimer's disease, in both of which nerves have been significantly damaged or lost. Conversely, you might find a huge brain tumor that has been growing for some time at this stage in the timeline. The black line shows that as disease progresses, there will be a significant loss of tissue. But while the disease diagnosis becomes more certain, the possibilities of saving the remaining tissue through therapy are much reduced. So the clinical treatment dilemma is when to be sure that you need to treat an individual by assuring the diagnosis, but still having a window of opportunity in which to do something to stop or slow the disease course. In this slide you see a cartoon of the scientific hypothesis underlying our clinical treatment paradigm. This is that a specific cell population, monocytes, will be isolated from the blood of an individual, that these cells will be specifically activated through cell culture, in which we will induce the desired functional properties of these cells, which will then be adoptively transferred back into the individual as part of their treatment. So be using a person's own cells to help cure themselves. So the question arises, why use monocytes or macrophages? In this slide you see a cross-section of the central nervous system of a mouse suffering from multiple sclerosis disease. All the brown spots are immune cells that have been specifically stained with antibodies. In the lower panel you see T cells, and there are a few of them scattered around the section. However, in the upper picture you see staining for macrophages, and there are hundreds of them invading the central nervous system tissue. It is their activity that leads to tissue destruction, as they have a pro-inflammatory phenotype in this setting, and that is why we consider that taming the actions of these monocytes macrophages is the key to efficient immunotherapy. In the next slide, a cartoon is presented of the underlying basis for the immunotherapy. On the right-hand side, you will see the immunological status of normal, healthy individuals, be they people or animals with a given genetic predisposition. There is a balance of black pro-inflammatory and white anti-inflammatory immune responses, which is symbolized by the yin-yang symbol. When one side is activated, the other side will restore balance. However, in the left-hand panel, you will see that the yin-yang is no longer balanced. In this case, the black dominates, indicating excessive pro-inflammatory immune activity, which is the hallmark of, for example, autoimmune diseases. The reverse scenario is true of cancer pathologies, where the white anti-inflammatory portion dominates. So in simplistic terms, our therapeutic approach is to restore the yin-yang by adding back the activated cells that are seemingly deficient in function or missing in the disease state. This means that we would adoptively transfer anti-inflammatory activated cells, the white ones, in the setting of autoimmunity and transplantation, while pro-inflammatory macrophages, the black ones, would be used in settings of cancer. In this slide we see the concept of an imbalanced immune system. Here you see a seesaw which is heavily burdened by pro-inflammatory red macrophages on the left. If one would increase the numbers of the blue immunoregulatory macrophages with associated production of IL-10 and TGF-beta on the right, then the immunological balance could be restored. This is the basic concept underlying our therapy, the giving back of high numbers of pre-activated macrophages with the correct activation state for the disease setting at the site of inflammation. This means that immunoregulatory blue macrophages are required in settings of autoimmunity such as multiple sclerosis and type 1 diabetes, but the red cells are required for treatment of cancer. So to test this hypothesis, we use experimental rodent models of disease. We grow bone marrow-derived macrophages, and we specifically activate them before adoptive transfer into the animal. In this slide, you see a cartoon depicting how we have developed our stimulation protocols, this slide representing about 3 years' work in the lab. We tested a variety of stimulants in different combinations, assessing the levels and content of physical markers on the activated cells, the levels and content of secreted molecules, as well as, most importantly, assessing the functional activities of the stimulated cells. We then tested the transfer system in our experimental models, the result being optimized protocols for each of these stages. In this slide you see a depiction of the type of experimental testing we do in vitro, as we use cell culture systems as much as possible. So the types of questions we address are: what happens if you apply the different stimuli simultaneously or sequentially? Are the activation states stable or are they modified? On the top left you see that. On the top right, what effect do these activated macrophages have on the effector functions of T cells? Can we modulate their activity? And in the bottom panel, what happens if you mix differentially activated cells? Which activation phenotype dominates, and how many cells do you need of one type to inhibit the activity of the other? In this last slide 11, you see the types of experimental disease conditions in which we test our myeloid cell therapy. These include diseases of the central nervous system, such as Alzheimer's disease, multiple sclerosis and brain tumors, as well as other autoimmune diseases such as rheumatoid arthritis and type 1 diabetes. In all these conditions, there is an inflammatory imbalance that we believe could be addressed using our myeloid therapy. I hope that you have enjoyed this presentation, and please check out the additional webcasts for specific diseases that explain in more detail how this will function mechanistically in these different disease states.